You're listening to the Animal Free Labcast, the podcast that puts humans, not animals, at the heart of medical research. Welcome to the Animal Free Labcast, everyone. I am back this week again with my new co-host, Dr. Gemma Davies. Welcome, Gemma. It's good to see
Carla.
you again.
Nice to see you again.
So tell us what's happening in today's episode.
So today we're going to be talking about the latest report from the government on the use of animals in experiments. We're also going to be talking about World Cancer Day, which has happened earlier this week on the 4th of February. And we've also got a special guest with us today, which is Dr. Cinzia Allegrucci, who is one of our breast cancer researchers at the University of Nottingham.
Brilliant. That would be great. Really interesting to hear from Cinzia. So before we get started, Gemma, tell us about the poll results from last week.
Yeah, so if you remember, the question was, would you support the government in redirecting funding from animal experiments into animal-free research? And it was very simple, yes and no. And quite unsurprisingly, the overwhelming majority was yes. There was only one person that actually said no.
Oh, there's always one, isn't there?
There's always one. So, next time maybe we'll ask a bit more of a divisive question, see if we get some tricky responses. Yeah. So thank you for everyone for voting.
Yes, thanks so much for doing that. And did we get any comments from listeners?
So we had a question come in from a lady called Ola in Northamptonshire.
Hi, my name's Ola. I wanted to ask, what are the biggest challenges still facing herpes law, and how can we as supporters help push it forward?
That's a great question. Thank you so much, Ola. So the biggest challenge at the moment is getting time in Parliament to debate the issue. There are obviously so many issues that need parliamentary time, and there's loads of good causes that are on MPs' agendas. But one thing that we know is that MPs listen to their constituents. So the best thing that you can do is to email your MP and ask them to support the campaign. You know, it would be great if they would speak in debates, if they would ask questions in Parliament, or even host an event for herpes law. So, and if you feel comfortable, then requesting a meeting with them is also really great. If they get to actually meet you face to face and to hear from you directly about why this is important to you, they're really going to sit up and take notice. We can send briefings to help you that you can give them, and, you know, with speaking points. So please get in touch with us if that's something that you would like to do. And we've got loads of tools on our website that make it really easy for you to email your MP and ask them to get involved.
Fantastic. So much that everyone can do to push this forward. So we need to make sure that MPs are aware that this is something really important to the public. Absolutely.
It's something that, we need everyone to do their bit. That's the only way that it's going to happen.
So what's grabbed your attention this week, Carla?
So I've been thinking about Chinese New Year, which was last week on the 29th of January. And it's now the year of the snake, as people might know. And the problem that I have doing this job is that I go down a little bit of a rabbit hole when I hear about animals, because I start thinking, are they used in experiments, they're used in medical research? And I found that snakes are used. And the ironic thing is that the very thing that should keep snakes safe from predators, their venom, is actually the reason that they're being used in medical research. So what I found is that snake venom is being used in treatments and being tested as, potential drug trials for diseases like heart failure and cancer, Alzheimer's, Parkinson's, autoimmune, inflammatory disorders and blood clotting. So absolutely loads of different diseases that they're being used for. And the way this happens is that they're milked for their venom. That's what it's called. So the snakes are forced to inhale carbon dioxide, and that knocks them out. And then their venom glands are massaged to extract the venom, which does not sound particularly pleasant for them at all. And I think some of the worst things is that they're kept in captivity for their entire lives. They're going to be used repeatedly for this milking process, and they're often caged on their own. I don't know very much about snakes. I've never kind of shared my life with a snake, but I understand from the research that I was doing is that some snakes are actually really social animals. So being kept in captivity on your own, I think would be, really devastating experience. So, yeah, that's what I found out, Gemma, which was all just, really upsetting. But it got me thinking about, well, what are the alternatives? There must be something that clever scientists, the people that we know and, and fund, must be doing something that means that we can move away from using snake venom.
Yeah, so this is actually a really interesting example of a key area of drug development where we can take inspiration from nature and then use that information to build and create new, better synthetic versions that we can use as drugs and therapies for human diseases. So around a third of drugs have actually been inspired by nature. And some really good examples are things like Taxols. So the brand name paclitaxel is a really widely used cancer drug. And that was developed from the bark of a tree, a Pacific yew tree. And statins as well, which are used to treat sort of high cholesterol, are inspired by a molecule from a fungus, which has been sort of developed into a drug. So the reason that snake venom is used in drug development is that venom works by targeting receptors and channels on the surface of cells, which are the same targets that we use in treatments for many diseases. So snake venoms are made up of a really complex cocktail of pharmacologically active components like proteins and enzymes, but it's thought that they could contain up to anywhere around 500 different active compounds.
Goodness.
But the problem is that venoms haven't been well studied because of the difficulties in obtaining the venom. As you described, it's unethical, it's difficult and it's expensive, and it just wouldn't be feasible to use actual snake-derived venom in actual treatments for patients. And also, as with all products that come from animals, they're variable and they contain non-specific molecules. So every animal is different and they could produce a slightly different venom, which means that it's not safe to use in a human because we don't exactly know what's in it. So we need to really consider what else we can do. We need ethical and reliable alternatives. And there's lots of ways that we can take inspiration from snake venom and other elements of nature to use this information in ways that do align more closely with human biology and modern science. So, for example, we can use computer-aided drug design to model the structures of venom toxins, and then we can study their interactions with human proteins. So we can then combine this with the emerging fields of venomics or anti-venomics.
Venomics.
Yeah, it's a great name, isn't it? And that, combined with bioinformatics, we can use these complex analysis to really understand the composition of snake venoms at a molecular level and then design targeted therapies based off that are actually going to be really effective and safe treatments for patients.
Oh, that sounds super fancy. So it's all about taking inspiration from nature rather than from exploiting nature and her animals.
Exactly.
That sounds like a much better approach.
It is. It's really the modern cutting edge way forward. And, it just removes the need to use animals.
Fantastic. So Gemma, you said you're going to talk a little bit about the government's latest report on animal experiments. So tell us about that. So I guess I'll just sort of preface that by saying that the government every year publishes reports into the number of animals that are used in experiments. It's actually the Home Office that licenses animal experiments. So this is kind of what's being sanctioned, what researchers are allowed to do.
Exactly.
Tell us about the report and what it's kind of telling us.
So this is a report that comes from a part of the Home Office that regulates the animal use licences. It's called the Animals in Science Regulation Unit. And they basically are responsible for checking that everybody who has an animal use licence is complying to the terms of that licence and looking after the animals in their care properly. So the report actually makes for a really difficult read because it gives quite specific information about situations where the licence conditions have been breached. So some animals have experienced adverse welfare or even died through negligence and lack of due care and attention.
So that's over and above what it was already kind of licensed that they could do to the animals, which,
exactly.
I would consider already far too cruel. So This is over and above that. They're breaching the licenses and things are going wrong, even more wrong.
Exactly. Okay. So there is a lot more information about this on our website. We've got quite a detailed report if you are interested to read more about this. But, like I said, it is quite upsetting. So, maybe not everybody wants to know about that. But some of the key points to really take away from this is that in 2023, there was 169 cases of non-compliance, which involved almost 155,000 animals, which is an 864% increase from 2022. So that's
goodness,
huge.
something's gone really wrong.
Yeah. And 78% of those cases were related to a failure to provide appropriate care, which resulted in the unnecessary deaths of at least 450 mice, rats and fish. And even more shocking from that is that 18 cases were related to a failure to provide food and water. And many of those cases were for several days, resulting in the deaths of some animals. So,
That's really difficult to hear, isn't it?
It is, isn't it?
Yeah.
Other cases involve things like animals were put into transportation boxes or bags for days and forgotten about and found dead later on. Other animals drowned in their cages because of malfunctions with watering systems, or they got stuck in parts of their cages. Really simple things that should have been picked up through regular monitoring and general care for these animals.
Well, that really highlights why animal-free research and all of the brilliant technologies are so important, because it just removes this horrible element from research right from the start.
That's it. It kind of really brings forward the point that the best way to protect these animals from being in these situations is to stop them being there in the first place. We have so many different ways that we can do this research without involving animals. But it's just, the fact that they are there and then they're experiencing these conditions outside of the, horrible experiments that they are going through is really upsetting. So as much as it's difficult to talk about, I think it is really important to bring this forward just so everyone is aware of the reality of the situation.
Absolutely, because it can feel very far away, can't it? It can feel like there's a lot of secrecy. So I do think, we have got a duty to kind of shine a light on what's happening, however hard that is. So, yeah. If people want to kind of dive into that more, as Gemma says, there's more information on our website. So earlier this week, it was World Cancer Day. That's on Wednesday, the 4th of February. So we really wanted to highlight some of the brilliant animal-free research that we're doing in the area of cancer. And it's such a big issue. I mean, everybody's lives have been touched by cancer in one way or another. Just some shocking stats I'm sure people will be aware of. 1 in 2 people will get cancer in their lifetime. It's just staggering. And Macmillan estimates that there are currently 3.5 billion people living with cancer in the UK. And Cancer Research UK say that cancer could add £14.4 billion to the UK's health spending every year. So it's just absolutely devastating kind of impact on people's lives, on the economy, on the health service, on the NHS. And sadly, animal experiments are still used as the sort of gold standard in cancer research. And it's one of the biggest areas of animal use, according to the government's stats for last year. But what we know is that researchers can cure cancer in mice. They've been able to do that for a really long time. But there are still so many types of cancer that we can't treat successfully, that we don't have good treatments for, that the outcomes, the prognosis is still bad. So we've got a lot of unanswered questions around cancer. So we really believe it's time to take a different approach and a more human-relevant, human-focused, animal-free approach. So we're really proud that we've been funding cancer research in an animal-free way for many years now. And we're going to be speaking to one of those brilliant researchers now. So Gemma, over to you.
Yeah, absolutely. Well, we're really pleased to welcome Dr. Cinzia Allegrucci, who is an Associate Professor in Cancer and Stem Cell Biology at the University of Nottingham. So welcome, Cinzia. Thank you for joining us. Would you like to introduce yourself a little bit to our listeners?
So, yes, my name is Cinzia. As you say, I'm an associate professor at the University of Nottingham, and my lab really works on breast cancer. So my background, I'm a chemist and pharmacist by training, but I worked in breast cancer for many years now. And we do, tackle this type of problem or disease by many different angles, including, developing models for replacing mice, mostly, in the research.
That's fantastic. So could you tell us what you are researching and talk to us a bit about triple-negative breast cancer and why it's such a difficult area to research and so difficult to treat?
We are focusing on breast cancer research, but we are particularly interested in triple-negative breast cancer. So triple-negative breast cancer is one type of breast cancer that accounts for about 15-20% of all cases. However, this type of breast cancer is most prevalent in some genetic backgrounds, especially in African Caribbean women, where this prevalence and incidence is much, much higher. We don't really understand why this is the case, because most of the genetic studies that we can access, in terms of retrieving data and look at genes, are actually from Western Europeans populations. So therefore, we are very interested to look at all the different genetic components that drive this disease. And why it's very important is because so although, the numbers are not as big, as I said, is only about 15-20%, unfortunately, this cancer type is very difficult to cure. So it's got a very poor prognosis. So patients are affected tend to be younger, and they are not really included generally in the screen. So, for example, in the UK, in the NHS screen. And they present very early age. It could be as early as in their 20s, 30s, and 40s. So obviously, it's a devastating disease. And because the screening is not available, normally they are detected at a very late stage of the disease. And therefore, a higher grade, most are metastatic. So therefore, it's very bad news for the patients.
It sounds like it's something that can happen to really quite young women.
Yes, it does. So as I say, why that is the case, we're not really sure, but the incidence of triple negative breast cancer, even in Western, populations, is increasing. So we think there might be a link with maybe obesity or other environmental factors that predispose to this type of disease. But still, there is so much more research that needs to be done. One important aspect that I haven't touched for triple negative breast cancer that makes this cancer also very difficult to cure is the fact that current targeted therapies, so the ones, for example, that target the receptors that we can screen for in breast cancer, like estrogen receptor, progesterone receptor, or HER2 receptors, are not present. So therefore, all the drugs that we would use normally as a first-line treatment for this type of cancers cannot really be used. So therefore, there is a very much a lack of therapeutic opportunities, and most of the patients are still treated with chemotherapy, aggressive chemotherapy, at the test go, and surgery. So therefore, combined with the early age, the high grade, the very aggressive tumor types, the lack of therapies makes this disease very, very, very difficult. And that's why we're so interested basically in understanding more about the disease.
So Cinzia, tell us about your research.
Okay, so what we are doing in the lab is to develop a model, a 3D model, which we make from induced pluripotent stem cells of the mammary gland. So what we do is to take normal cells from the breast. We revert them to embryonic-like stem cells, also called induced pluripotent stem cells, and then we differentiate into the mammary gland organoid. So it's very rudimental mammary structure that resemble really the gland as it is in a patient. And also what we do is to induce then with gene editing some mutations in these cells so that we, as I said before, we can study the impact of this mutation in driving disease forward. By doing this in this model, we can study both cancer initiation, but also cancer progression, as this mutation will lead to advanced breast cancer. By using this model, obviously in the lab, we can do all of this in the Petri dish, which means that we're not needing mice to study the disease as we normally would have done in the past.
So one of the really important parts of your project as well, Cinzia, is, you were just saying then that you use a lot of different factors and things to get these stem cells to turn into the type of cell that you want. But everything you use is completely animal free, isn't it?
Yeah, absolutely. So through the years, obviously developing this model has taken a step-by-step approach. And I think, we are now at the stage where we can test and drive a completely animal-free system, which is very important, from ethical issues, but also is very important for drug testing, because that level of animal-free components it is very important to allow this model to be very reproducible, which is essential, for a drug screening sort of model or platform. So we at the moment, and thanks to the funding of Animal Free Research UK, we're testing different matrices which are completely synthetic. So we avoid using collagen and matrigel, which are animal derived. So we use peptide hydrogels that we actually obtain from biotech companies here in Nottingham. And then use all recombinant growth factors and protein and completely animal-free media for everything that is required for the cell culture of these organoids. In addition, we are trying now to step forward to use animal-free antibodies for the characterization also of these organoids that we need to stain to make sure that they produce the cells that we're expecting them to do. So this is the first time for us to use animal-free antibodies. And I'm very excited, that I actually found many companies that are now providing this protein, these antibodies, which means that, there is a very clear commitment from, product companies and industry really to go that way, which is really important.
So why is animal-free research going to lead us towards better treatments and outcomes for patients?
So our research, we like to believe is very important, going forward for patients and patient outcome, because models like ours, can really drive forward this so-called precision medicine. So by looking at the individual mutations that people carry, so we hopefully can stratify better patients, so to allow the development of specific treatment for specific patients. However, as I said, previously, it's very important that the models that we use are refined as possible to allow, good drug development. So therefore, the animal-free approach is very important, for this reason. In addition, I think, for patients, it's very important in their cancer journey to know that every research that is done, for them is ethically sound. So at all levels, so not just, for patient involvement, donating their, tissues for research, but also for the use of animals. We all have animals that we care for, and I think patients are really sensitive, to this issue. And I think, they are very driven by the fact that I think, so they're really interested in understanding that the research that we do is really mindful of not causing unnecessary suffering in animals, if at all possible. So, I don't think any patients will, think that the, method justifies the means, because, obviously research is important for them, but, not at the cost probably of animal welfare.
And presumably, Cinzia, you're talking about research eventually becoming personalised, so to the particular patient. So presumably, you want to make sure as much of your research is done in kind of human conditions as possible, so you can get as close to what's actually going on in patients.
Absolutely. This is a very important aspect. We know the attrition rate, for cancer drugs is very, very high. So I think, that comes with the huge costs, both for, the public, in terms of tax paying, but also, for pharmaceutical industry, which, obviously the more losses they make, the less they invest in future drugs. So I think most of this attrition rate comes from the fact that we are using, models that are not representative of the human disease. And we keep doing this despite we all understand that that is the case, because there are no better models that we can actually use. Of course, animal models are important ethically because, we can't test new drugs in patients directly. But if we can avoid animal models altogether by using relevant, clinically relevant models for human disease using human cells, I think that is a win-win situation.
Yeah, thank you so much for everything that you're doing, Cinzia, really at being at the cutting edge of animal-free breast cancer research. You are such an inspiration.
Thank you very much to you and to the supporter and to the charity Animal Free Research UK for the support on our research.
So Gemma, we've got a cancer research related poll question for this week, haven't we?
We have. The question this week is, do you think it's time for cancer researchers to embrace non-animal methods in their work? And again, the answer is yes or no. And you can vote on Facebook, Instagram, X and Carla's Substack, the Animal Free Lab Stack, and we'll discuss the results in the next episode.
Just repeat that question again one more time for everyone.
It's, do you think it's time for cancer researchers to embrace non-animal methods in their work? Yes or no?
So please stay part of the conversation by reading my brand new companion blog on Substack. Just search for Animal Free Lab Stack to read and subscribe. You can leave us a comment there and you can vote on the poll there or on one of Animal Free Research UK's many socials. Gemma, remind everybody how they can leave a message if they'd like to do that so that we can feature you in next week's, in the next fortnight's episode.
Yep, so if you'd like to tell us your thoughts about the poll or anything else about this episode, you can leave us a message on 0208 054 96 95. So again, that's 0208 054 96 95.
Please do that because we love hearing from you and we'd love to play some of your messages. Well, thank you so much for listening, everybody. We'll be back in a fortnight when we are going to be talking about heart disease because February, of course, is Heart Month. So until next time, take care and we'll see you soon.
And thanks for listening. See you next time.
You've been listening to the Animal Free Labcast, the podcast that puts humans, not animals, at the heart of medical research. If you want to find out about more ways to move to a world of animal-free research, then head to our website, animalfreeresearchuk.org. And if you'd like to send us an audio message about the poll or anything else you've heard in the podcast, then leave us a voicemail on 0208 054 9695. That's 0208 054 9695. Thank you for listening.